This video is going to be covering transcription, which is a synthesis of RNA under DNA. The three phases of transcription are initiation, elongation, and termination. There are slight differences for transcription of eukaryotic and prokaryotic cells, but they result in the same end product. Differences include usage of different proteins, such as different versions of RNA polymerase, and the need of more helper proteins in prokaryotic transcription due to the process occurring out in the cytoplasm. The first step is called initiation, in which the DNA unzips for RNA polymerase. The second step is called elongation, where RNA polymerase reads a template strand and creates mRNA. The third step is called termination, where the RNA polymerase reaches a Rho protein, or sometimes known as the intrinsic termination, which causes the RNA polymerase to deactivate. DNA has the unique ability of not only creating complementary strands from the template strand, but also can create mRNA strands used to express the genetic information. The picture I drew is a DNA strand compared to the RNA strand that will be created if both were in the 3 to 5 direction. However, mRNA is flipped in the 5 to 3 direction because it is anti-parallel to the template DNA strand. The key difference between mRNA and the template DNA strand is the usage of uracil to replace thymine bases. RNA also uses a ribose sugar instead of a D-ribose sugar. Transcription process in eukaryotic cells involves the usage of RNA polymerase to carry out RNA synthesis almost entirely. RNA polymerase unzips DNA by removing hydrogen bonds between complementary bases and then using free RNA nucleotides in the nucleus to make a complementary anti-parallel strand. The mRNA chain is produced in the 5 to 3 direction despite RNA polymerase moving in the 3 to 5 direction in order to create the needed anti-parallel strand. The DNA strand is then rewinded and the RNA polymerase ends transcription when reaching a Rho protein, which blocks further movement. Splicing occurs in both processes of transcription no matter the type of cell, in order to remove the RNA sections that don't contribute to the coding. These not useful sections are called introns because they interfere with coding. Exons, on the other hand, are called exons because they express information needed to build a protein. These intron sections exist because most proteins only use around 400 amino acids, which is just a small section of the large mRNA chain. So this is where splicer proteins are needed. Splicing works by helper proteins called snRNPs, marking exon and intron borders, and then forming into proteins called spliceosomes with additional protein help. This then deattaches the introns and reattaches exons before deattaching from the strand. snRNPs are rather large, in fact almost the size of ribosomes, which are self-reliant in catalyzing introns from exon parts. An important note is that there's so much genetic variation in the living organism due to the ability of snRNPs to determine which section of genetic information to ' and what to throw away. Anyways, after splicing occurs, eukaryotic cells process mRNA into mature form by adding a 5' cap, which is a modified guanine amino acid with 3 phosphates, and a poly-A tail on the 3 end, which consists of 150 or so adenine nucleic acids. This addition of the caps facilitates transportation out of the nucleus, prevents degradation by hydrolytic enzymes, and helps ribosomes attach on the mRNA through the 5 cap. As discussed before, prokaryotic transcription is different from eukaryotic transcription due to the process occurring out in the cytoplasm. Initiation occurs from the DNA being opened up by the helicase enzyme that breaks down hydrogen bonds. Once the DNA is separated, the transcription is initialized by the operon on the promoter region. The presence of the repressible protein in the operon determines whether or not the RNA polymerase II enzyme can latch on. This repressible protein is created by the regulator gene upstream from the genetic material. There are 2 types of operons: the inducible operon, in which transcription takes place when repressible protein is removed off the operator, and repressible operator, where transcription normally takes place and repressor protein is used to stop transcription. That is it for transcription of prokaryotic and eukaryotic cells. I hope you have gleaned some information from this presentation, and bye.